You have heard a lot about DNA. What exactly is it? Specifically, what is it made of? Like most macromolecules, DNA is a polymer. That means it is many monomers, or small molecules, that have been covalently bonded together into a chain. Remember, mono means one for these individual molecules, whereas poly means many, signifying that they've been strung up.
Hey, Miss Day, do you remember which of the 4 macromolecules DNA is?
It's gotta be protein.
No. Okay, so DNA is a nucleic acid, and that's where it gets its name, deoxyribonucleic acid.
Oh, that makes sense. Okay.
But what I really can't remember is, what are the little monomers called that make it up?
Nucleotides. They're called nucleotides.
Oh, thank you. That sounds a lot like nucleic acids. That will help me remember. Thanks.
You may have remembered, just like Miss Day, that the monomers of nucleic acids are called nucleotides. What you probably didn't know is that nucleotides have 3 parts: a phosphate group, a 5-carbon pentagonal sugar called deoxyribose, which is where the D in DNA comes from, and then lastly, you know that DNA carries genetic code. The section of each nucleotide that carries the code is this part, called the nitrogenous base. And the nitrogenous base is so named because it contains a lot of nitrogen in it. The nucleotides are just the monomers. You have to string them up in order to make the polymer. So here's one of those nucleotides with its nitrogenous base, deoxyribose sugar, and phosphate group, covalently bonded with another, and then another, and then another in a chain. That's what makes the polymer, which is called deoxyribonucleic acid, or DNA for short. The individual nucleotides are strung together from phosphate group to deoxyribose sugar, to phosphate to deoxyribose sugar, and back and forth, creating a backbone of the molecule called the sugar-phosphate backbone. Each deoxyribose is then covalently bonded with a nitrogenous base. There are 4 nitrogenous bases, each with a slightly different structure. For example, thymine only has 1 ring, whereas adenine right here has 2 rings. Depending on the order of nitrogenous bases that occur inside the DNA molecule, that's what stores the code. Now, the previous diagram is a little bit misleading because, as Rosalind Franklin and Watson and Crick all found through their research, DNA actually comes in a double helix. So that is a helix with 2 strands running through it, rather than just the one. So the diagram we were just looking at looks like half of the molecule right here, but there really is a whole other half which is interacting with that half. So DNA is really a double-stranded helix. Now, how did these guys come to find it? Rosalind Franklin took a bunch of DNA and crystallized it, put it in a crystal, and then used a technique called X-ray crystallography, in which she shot radioactivity at that crystal and then collected on a photography plate how the radiation bounced off. And she's much more sophisticated and smart than I am. I don't really understand how to interpret this picture, but when she looked at it, she said, these X's right here represent that there must be grooves in this long molecule, such as the groove right here, a major groove that runs this way, and then a little minor groove between the strands right here. And so she said, all right, this molecule must be in the shape of a double helix. Then it was actually Watson and Crick who went in, took her data, and tried to construct a DNA model, like you can see in this picture, that would ensure that every single atom got the full number of electrons it needs to fill its outer shell. And the end result was our modern model of DNA. So why is it called a double helix? For one, there are 2 strands, this side and this side. And for every nucleotide in one, there is an opposing nucleotide on the other side. So this nucleotide, you can see phosphate group, deoxyribose sugar, and thymine nitrogenous base, is across from a nitrogenous base, a sugar which is hidden right in the corner here. So I just drew it in. And that's connected to a phosphate group. And the matching across the helix is not random. For example, here's a G across from a C. And down here, there's a G across from a C. And here there's a G across from a C. Why do G's and C's always pair together? Why do A's and T's always pair together? The nitrogenous bases have unique chemical characteristics that allow them to match with each other perfectly. To keep them straight, usually biochemists will sort nitrogenous bases by size. So adenine and guanine, both of which have 2 rings, are called purines. Those are the large ones. Whereas cytosine and thymine have a single ring, they are called pyrimidines. These 2 categories are easy to mix up, so to help me remember, I invented something. This is Adgua, the purine monster. You can see he's a purine because his eyes are made out of double rings rather than a single ring. See, Adgua, AD for adenine, Gua, that's for guanine. So anyway, A and G are purines, C and T are pyrimidines. Franklin's photograph showed that the helix must be smooth and continuous along its length, which made her think, well, there couldn't ever be 2 purines across from each other because that would be too wide, and there couldn't ever be 2 pyrimidines across from each other, that would be too narrow. In order for the width of the helix to be consistent, a purine would always have to come across from a pyrimidine. And as Watson and Crick later confirmed, that's how it happens. Purine nitrogenous bases are always across from pyrimidines. Watson and Crick then built on Rosalind Franklin's understanding by trying to figure out which purine matches with which pyrimidine. They knew that for the helix to hold itself together, the bases must hydrogen bond with each other. You already know that hydrogen bonds are weak attractions between partially oppositely charged molecules. So how did these bases get opposite charges? Well, Watson and Crick knew that oxygen is an electron hog and would therefore be partially negatively charged. They also knew that nitrogen is close to oxygen on the periodic table and thus does the same thing. Meanwhile, these hydrogen atoms are all left positive because the nearby nitrogens have been hogging their electrons. Now, when a guanine and a cytosine are across from each other, this positive charge matches up perfectly with this negative charge. This positive to this negative, negative to positive. Whereas the guanine wouldn't be able to match up with the thymine because then this negative charge would be right next to this negative charge and they'd be repelling each other. So these hydrogen bonding patterns causes the A always to hydrogen bond with the T and the G to always hydrogen bond with the C. Another memory trick. C and G fit together very well because their shapes are similar. Same with A and T. In conclusion, the DNA double helix is held together by 2 kinds of bonds. Strong covalent bonds between sugar and phosphate in the sugar-phosphate backbone, and weak hydrogen bonds that attach the A's to the T's and the G's to the C's across the middle of the helix.